Y traerlo un poco más acá.
No, voy tomando el video para la compu. Ahí está, ahí quedó. ¿Ahí va? Sí. Espera que actualizo el link en la página.
It's birthday. Oh, happy birthday! Then we have to sing. Yeah, we can find we almost in each course we have someone. Yeah, birthday. Yeah. You're the host. Reduce your $11.
Avisame, ¿te escuchas? ¿Cómo? Bueno, estamos... Good morning. Thank you all for coming, and thanks to the people following us online. I am Alberto Peña Ensembl. I have been coordinating this workshop today. Let me thank the Science and Technology Secretary, Dr. Federico Coluccio, and CDTIC Director, Daniel Pelosa, for hosting us and for making possible the broadcasting of this course today. Now let me introduce you to Bastital. She obtained her PhD in molecular biology from the University of Veterinary Medicine, Hanover, Germany. Okay.
Yeah.
She worked at the Federal Research Institute for Animal Health from Germany, the Ensembl Institute, and the University of Cambridge, both in UK. Astrid joined Ensembl as an outreach officer in August 2017. There she delivers workshops, creates training materials and help tips, manages social media and answers help desk queries, and help the development of new Ensembl tools.
Yeah. Good morning, everybody. Thank you for the introduction. Welcome to this course, including for the remote participants. I work for Ensembl. We are at the European Bioinformatics Institute, and this is near Cambridge in the UK, and we are part of Ensembl, so the European Molecular Biology Laboratory that has its headquarters in Heidelberg and different sites within Europe. This is our plan for today, rough plan for today. I will start with an introduction and talk about different genes and transcript views. We're going to have a coffee break around 10:30, and we will continue with variation. Then we have a lunch break for an hour, and in the afternoon we will do comparative genomics and biomart, and if we have enough time, maybe data upload as well. All of these modules have a similar structure. We start with a presentation where I will introduce the data or the type of tool and how we produce the data and process it. We will then have a demo where I will show things on the large screen live, and you can either follow on your own computers or just watch what I'm doing. And then we have exercises where you can try what you've learned yourself. Please do work with your neighbors and discuss these with each other. There are plenty of exercises for each module. I do not expect you to be able to do all of these, so you can pick and choose the ones you are most interested in, and I will walk around the room and help you with those as well. The materials for today's course. Are we okay with the technology? Yeah? Are we okay with the sound?
Yes.
Yeah. All materials for today's course can be found on this website, training.ensembl.org, and I believe you have been sent the link already. I will still go there because not everybody will have checked, probably. So this website is this one here, training.ensembl.org, and there's a link at the bottom left where you can find materials of all the workshops that we are doing. Come on. And this is ordered by date, so today's date is the 13th, and there's our workshop. And here you have got a presentation, a course book for the demos, a question book for the exercises, exercise answers, and also a plain text file, a copy sheet which we will use later today. All of these materials will stay there, so you can always download them in the future as well. Okay. I hope that by the end of today you will have learned what Ensembl is, what type of data you can explore in our browser, how to navigate the browser website, and then finally and importantly, where to find further help and documentation. There's a lot of it available online for free, and I will point you to that later. I'd like to ask a question to you. Who has used Ensembl before? Okay, a handful of people. That's fine. We start with the basics. I'd like to say that please do ask questions at any time. There aren't any stupid questions. I'm really happy to answer all the questions you have. And please just interrupt me when questions come up. So it's quite, it's a lot easier to answer them directly. Also interrupt me if I'm speaking too fast or if I say something that doesn't make sense, or if I use words that you don't understand. Okay? Good. Another thing to check is, I see some people don't have computers. Are you happy to share with your neighbor? Yeah? Because there is a spare one, I think, in the front. if you want to move, you can use that one. Okay, I'll start with the first presentation now to introduce you to Ensembl. I will cover in this session genome browsers in general and then Ensembl in particular. I will talk you through our release cycle that we use to update our data constantly. And finally, I will also give some background about genome assemblies because these are important for some of the views that we have in the browser. Okay, so we've been sequencing genomes for a while. The first one was finished in '77, and that was a bacteriophage done by Fred Sanger. The next big milestone was the finishing of the human genome sequence in 2004. And between those two milestones, we of course had different other genomes being finished, and later, in some cases, also multiple individuals of one species, such as for humans, where we have thousands of genomes now. The question is, why do we need a genome browser? My answer is this. This is loading. It's thinking for a moment. Okay. I need to check if we are still on the network. No, we are not. This seems fine. Okay, I think we dropped the internet connection there, maybe. Is anybody else having this problem? No? It's just me.
Sabri, ¿qué tipo de seguridad tiene la de ¿No te aparece ahí? ¿WPA será? Sí. ¿De TIC? A ver. Sí, se recuerda. No, no, es que sí, no,
Y se puede tocar lo mismo. No sé, no conozco. ¿Cómo
ahí, conectado.
¿Ahí conectado, no?
Sí, ¿eh?
Sí. Pero acá tenés que poner el proxy.
Ahí va el proxy. Ah, ahora ya.
Bueno, ¿cómo ya?
Good now?
Check.
Try. Yes, thank you very much. Thanks. Okay, sorry for the short disruption. So I was saying, why do we need a genome browser? This is my answer. This is how a genome looks like. A string of letters of A's, C's, T's, and G's. And we need some way of unlocking this code. This is where genome browsers come in. There are 3 different genome browsers around. I work for Ensembl, so I'm going to talk about Ensembl today, but I will not neglect to say that there's also the UCC genome browser and the genome data viewer in the US, and they do similar things. They just have different interfaces and tools, and it's up to you which one you prefer. Ensembl now is very comprehensive. We have genomes for more than 200 species, and this is linked. These are linked to 3 other types of data. We have variation data. We have comparative genomics data, so that's alignments, gene trees, and homologs. And we have a regulatory build as well. This data can be accessed by a variety of tools. We have a tool for data export, which is called Biomart, and we use this afternoon. We also have tools for data processing, including our variant effect predictor. Again, we're going to use this later. In addition to this, you can upload your own data to Ensembl in a variety of file formats. If you're interested in larger scale access and you can do some programming, you can use our APIs as well. And finally and importantly for me is that all of this is open source. So it's freely available for everybody to use. And this includes the whole genome databases on the FTP server and our code, which is on GitHub. You can access our databases on different scales. We are going to mainly use the browser and our tools today. So if you are looking at individual genes or variants one by one, you will use the browser. If you then move on to groups of genes, say you are more likely to use one of our tools, such as Biomart or the web or maybe the REST API. If you, however, want to do a whole genome analysis, you will have to use the FTP server or the Perl API or the MySQL databases. These 2 last methods here are certainly the most flexible ones and most powerful ones, but they're also the most complicated ones, and therefore I will not teach them today. But just be aware that these exist if you need them in the future. This phylogenetic tree now summarizes all the species that we have in Ensembl. They are mainly vertebrates, but if you are very eagle-eyed, you will be able to spot that there are some model organisms in here as well. At the top, these form the outgroup of this tree, and they're used for comparative purposes. We have got Drosophila, C. elegans, yeast, and Siona in Ensembl. In addition, there is a sister site which is called Ensembl Genomes, and this covers non-vertebrate species. This is organized into 5 mini sites which are color-coded for bacteria, fungi, invertebrate metazoa, plants, and protists. These websites work in a very similar way. If you can work Ensembl, you can work with Ensembl Genomes as well. And in fact, we are in the process of merging Ensembl genomes into Ensembl 2. Is there anybody here who is not working on vertebrates? What do you work on?
Plants.
Okay.
Anti-education.
I don't. Yeah, I know. I'm thinking we don't have this genome. I think we have a variety of plants, but not this one.
The criteria to include the genome sequence, for example, eucalyptus sequence, but it's not included in what is the criteria to include or not?
There needs to be an annotated genome available in INSPC, so ENA or GenBank. And then you you just write to us and say, I would like to see this genome in Ensembl, and we check the quality of the assembly and the annotation, and if it's good enough, we will include it. So yeah, you may want to do that. Then somebody else, what are you working on? Also non-vertebrates?
Working on bacteria.
Bacteria. Also bacteria. Anybody else?
Plants.
Fungi, plants, also plants, okay. Cool, so we have a mix here. Excellent. I included exercises for all different taxa, so you can pick the ones that you work with for the exercises. For the demos, I will stick to human. But as I said, the websites work in a very similar way. Okay. The main difference between Ensembl and Ensembl genomes now is the annotation. I've just said, so if you want to have a genome in Ensembl genomes, plants for example, there needs to be the genome and the annotation available, because we do not annotate the genomes. So for Ensembl genomes, annotation is done by members of the scientific community, and we import that data along with the genome. For Ensembl species, we annotate the genomes ourselves. So that is the main difference there. To keep up with all the new data that comes along, we work on a release cycle, and this is approximately 3 months. The release before the current one was Ensembl 95, and that came out in January. And at that stage, the databases are frozen, and we stopped the development of new web interfaces and additions. And then we work behind the scenes to work towards the new release. And what happens there is that we import new genome assemblies, we make software updates, we update our gene sets of existing species, we then run the comparative genomics pipelines with the new genes and the new genomes. We also update variation and regulation data. And finally, we update interfaces or include new interfaces. And then we put out a new release, and the current one is 96, and that came out in April. And now we work towards 97. So that's our release cycle. This is important for you because a lot of data analysis takes longer than 3 months. So what you want to do is you write down this release number you started your data analysis with, and then that enables you to continue it later, because we have archive sites going back for years and years, so you can always go back to older releases there. Okay. Finally, in this session, I go over genome assemblies. I'm sure some of you will have done this already. If you sequence a genome, you generate short sequencing reads, and the length depends on the technology. And what you need to do is you need to put them together into a contiguous sequence. And the way this works is a little bit like a jigsaw puzzle, so you need to find the overlaps between these reads, and then you trim the trim things away, and then you end up with this contiguous sequence or contig. And this is then where a little bit of genome browser jargon comes in to name the different steps in this process. A lot of the reference genomes were sequenced by cloning them into bacterial artificial chromosomes, so that's like 15, 20 years ago. And we then can view these clones in our browser. And this is an example of 4 different clones shown as red blocks. And then the data that we show in the browser as these clones is what we call the typepath track. A track is just genome browser jargon, so that's a word that describes. So our clones are this typepath track. If we then trim away the overlaps between these clones, we get contigs. We show them in alternating light and dark blue, and such as here. So we have got 4 contigs here, and then we call a run of contigs without any gaps a scaffold. And then for many genomes, we then have used genetic maps to assemble these couples into complete chromosomes. That includes human, mouse, and other genomes. So yeah, the important thing to note about human and some other reference genomes is that these are not genomes from one individual. There are actually composite genomes which were derived from the DNA of different people. In this example here, I've got a chromosome where 4 people contributed DNA, and then their DNA was derived to get these 4 different contigs of the chromosome. So yeah, just keep that in mind. The reference genome is not in any person. Talking about the human genome assemblies, these have been updated over time as well. The most up-to-date version, and the one that we have in Ensembl, is called GRCH38, which stands for Genome Reference Consortium Human 38. This does not have any gaps, and we update it every 3 months. We do know that, in particular in the clinical community, people still work with GRCH37 as well, which has 250 gaps. Because it's one that a lot of people still work with, we have an archive site for this, which is called grch37.ensembl.org, and we make limited updates to it as well, and we support that too. If you read the older literature, you may come across NCBR 36, which has a lot of gaps. We do not longer support it, but you can go to our archive sites, which are shown in brown, and to have access to it. Yeah, I think that's it for the assembly step. Any questions so far? Yeah.
When the human genome was sequenced, there was a private project and a public project, right?
Yes.
How is it related to these 4 people human genome that is in Ensembl?
So this is just a made-up example. It's not only 4 people.
Okay.
The human genome was, as you said, it was a competition between 2 collaborative projects, and in reality, so we don't know the whole details, but we know that the majority, so the part that was done in the US, is actually an African American male. That's what we know. but we don't know which individual context came from what person. So it was just cloned and it was all anonymized, and the clones are just labeled with a combination of letters and numbers. More questions? Okay. Then we're gonna have a look at this now in the demo, which is in your course book on page 6 to 12. And as I said, you can follow on your own computers or just watch what I'm doing here. If you want to find the computer, again, there's one in the front. Okay. I'm going to leave my presentation and I will go to ensembl.org, which looks like this. Can everybody see this in the back, or is this too small? No? Okay, cool. So on the homepage, you have at the top you have this blue header with a logo and then access to tools, documentation, and other things, plus a search box. This top header is always visible in any page in Ensembl, and you can access these things anywhere. If you're ever lost, you click on the logo, which will take you back to the homepage. On the homepage, you have information about the release on the right-hand side. You have a tool section at the top left, and then you have a search box, a search section here. Here you will have to select the species from the drop-down menu. We have some favorites set, and then you type in what you search for. As you can see from the examples underneath, our search boxes are very powerful. You can put in genes, you can put in locations, variant identifiers, and also things like phenotypes. Then we have a genome section here, again with a drop-down menu. There's a link to a table with all species in Ensembl here, and we have some favorites too. And towards the bottom, there's one thing I want to point out here, which is this tiny link view in archive site. So this is where you can access the archives at the bottom right. This gives you a pop-up window with all the archives we have, their dates, their genome assembly version, and you can link to those. I will close this again because I don't want to go to an archive site. To close any of the windows in Ensembl, you can use the tick mark at the top right and also just click outside the window. Okay, so now I will visit the human genome. So I click on my favorite here. And on this page at the top, you can see your search box again, and just below the assembly name, plus its accession number in brackets. And then we have 4 boxes which take us to example pages to our different data types. So for gene annotation, variation, comparative genomics, and regulation, all linked with example pages. If you want to find out more about the genome and the assembly, you go to more information and statistics at the top left. And here you can see a text description of this human assembly and how the annotation was done on the left-hand side, including about patches. Patches are updates to the genome which are added after one assembly version. So you can see at the top it says human GRCH38.P12. That means patch 12. Yeah, and there's information about how the genome build was done, including a PDF file with the details here. On the right-hand side, you have got summary statistics in a table, again with dates, but also things like gene counts towards the bottom. You also have a drop-down menu on this page, here under Other Assemblies, which takes you to archive pages. You can choose from GRCH 37 and Ensembl 36. I would choose GRCH 37 and click on Go. And this takes me to this GRCH 37 page, which looks very similar, except that it's now light purple and the logo has changed. What has also changed is the address bar in the browser, so our www is now GRCH 37, and this is in fact a shortcut to any archive site. You could say, for example, e94.ensembl.org, and that takes you to the archive site for Ensembl 94. Archive sites are shown in brown. If you are today, during the day, at any point in a brown web page, you are in an archive site and you probably don't want to be there. Okay, so that's Ensembl. I'll go back to the home page and I will now visit Ensembl genomes. So this is found at ensemblgenomes.org. Here on the left we have archive sites. Don't use these links. The links you want to use are on the right. So they are split by bacteria, fungi, metazoa, plants, and protists. You can access these pages here on the right, but also in the top header. So going to fungi takes you to the brown fungi page. Plants screen, as you would expect. You can also change from one side into the other by using this down arrow next to the logo. So you could go back, say I want to go to bacteria. I've gone to bacteria now because this is the site which works a little bit differently. The reason is that we have 44,000 genomes, bacterial genomes in Ensembl, and therefore you cannot search for gene directly. So you need to search for your genome first, so your strain or species, and then for your gene. And I will do this now. I will search for Streptococcus pyogenes by typing this into the genome search box, and it will give me some options. And here I have to select the strain I'm interested in. I will go for this one here, AA216. And now I'm in the homepage of that strain, which has a link to the taxonomy here. And I can search for the gene now. So these are the two steps you have to do for bacteria, really. Otherwise, it's just the database is just too large to search for a gene directly. Yeah, so that's everything I wanted to show for the homepage and the species. Questions on this demo? No? Okay. If there aren't any questions, then we have some exercises now, which are in your question book on page 3. And as I said, there is an answer book as well. Please feel free to use that if it helps. And yeah, I will walk around the room and help you. Yeah, that's true. Exactly.
Always start is you find all the
Yes, so I'll just go back to the previous year, just writing that. Okay.
always appears here on the values for that aspect.
Okay, so how do we get on? Who would like more time for exercise 1 or 2? Okay. See a lot of people working on the bacteria already, which is great. We give it a couple of more minutes. Yes. Yeah. Okay. Are there any questions on the exercises? Do I need to show anything? No? I think I will do a second demo now before we break for coffee. Does that sound alright? In this demo, we are going to look at one region of the human genome, which is on chromosome 4, and it's a region shown in red up there. You have to write down regions in Ensembl in a certain way, otherwise it can't read it, and it will try to read it as text. So you need to start with the chromosome, here 4, and then a colon, start position minus end position. No commas, no spaces, no dots. It will not be able to read it. And we will take this region and add some data to it. This demo is in your course book on page 12 to 8, and as before you can follow. I will copy this region. I will go to my browser, select human, and paste it in, and click go. Ensembl recognized this as a region, so we are now have been taken directly to the what we call the location tab with the region detail view. You can see it's open. Location tab is open on the left. As we start working with more and more other features, we open more tabs. So now our menu on the left here is specific to location views. The first thing I want to point out on this page, though, is this question mark here. This opens, if you click on it, a page-specific help opens. In this case, this view is explained in great detail. It includes a YouTube video at the bottom here, where a previous colleague explains this page. We have this for quite a few pages, but not for all, obviously. So I will go back here and say, in the help pages we also have FAQs. We have a glossary and finally a form to contact us at help desk. Here you can write a message. We would like to ask you to send a link to the page as well, or send a screenshot or something which you can attach, and then provide us with your email. We will not spam you, and we will try to get back to you within 1 or 2 working days. So this is one way of getting in touch. Okay, I will close this now and go to this page. There are 3 pictures on this page. You zoom in from this chromosome view into the next, and then from there into the next, which is a highly detailed, customizable view. To go through these, at the top in the chromosome view, you have the cytogenetic bands shown in black, white, and gray. This red box here represents where we are at the moment, and these marks here are the alternative sequences, the patches. You can move around here by dragging a box with your mouse and then say jump to region, and you could likewise mark a region. I will close this though. So this second picture here now is a 1 megabase overview around this red box, and this is where you start seeing tracks. So there's the first track, for example, here for the chromosome bands, which have labels, and then just below there we have contigs. Next track is starting to look at genes, where each of these blocks here is a gene, and there's a legend with the color codes at the bottom of the page, but we will come back to these genes later. Again, you can move around here by dragging boxes and say jump to region, or you could also change your tool at the top right to this double arrow, and that's then like a drag tool, so you can drag this with your mouse and move to the left or right. And when you do this, you will see that this third picture now has grayed out, and you will be asked to whether you want to update it or go back to the reset it, go back to the original setting. This is still loading. I'm gonna refresh this. The internet is fine, it's just taking a moment to load. If you can see this on your screens already, the third picture is a split, and yeah, this. Yeah, I'll wait for a moment. Until this loads. I point out a couple of other things. So here, now it has loaded, of course. So my third picture now contains a lot of details about this region. Just for orientation, you have your track with your contigs here in blue. Everything above that track is on the forward strand, so these are transcripts of forward stranded genes, and then everything below is on the reverse strand. There are some such as variants, which are shown at the bottom as well. We have a legend at the bottom that explains the colors, for example for the genes, which are shown in red, yellow or green. I come back to this later. What you can do with this picture is you can customize it and add data you're interested in. For this, you have to use this button here on the left-hand side, configure this page. We're going to use this over and over again, so it's a very important one. So if you click on that for the first time, you will see in the menu all the different tracks that we have. They are organized by things like sequence and assembly, genes and transcripts, and so on. And these numbers here represents the tracks that you have switched on and how many are there in total. So here we have 3 out of 18 switched on. You can navigate through this menu, but you can also use the search box at the top left to find the data you're interested in. So I will search for a protein track. I will type in protein here, and it brings up the options on the right hand side. This one is already switched on, and I will add a track here from UniProt. So proteins memo from UniProt. I will check this box, and I'm asked how I want to show this. I will say for my track style labels, and then I close this window, and my protein track loads onto this view. Once you've done customization like this, you can save this picture. You can use this icon here, export this image to download the picture in a variety of different formats. You can also share a link to the page with a colleague or save a link. For this, you use the share this page button on the left hand side, which creates a unique link to your page, including all the customization you have done, and the release number, so you can copy it and send it to somebody. If you use the link from the address bar of your browser, this will only include the location and not the customization you have done. So I'll close this. Now my protein track has loaded. You can see proteins in yellow with their labels. You can show tracks in a different way by clicking on the track name and then the cog wheel and selecting a different option. If you go for normal, for example, you won't be seeing the labels for the proteins. Okay, while this is loading, I will point out that there's also a zoom bar here at the top, so you can zoom in and zoom out. I personally find it a lot easier to drag boxes with my mouse, as I've shown before, and then say jump to region. It's more precise than using this bar here. Okay, so my protein track doesn't have any labels now anymore. You can move tracks around by grabbing them on the left-hand side on this colored bar and moving them around. You can also delete them by clicking on the name and then the X. Yeah, so you can really customize this page. It's important to reset it to the original configuration from time to time, especially in a workshop, because it can become quite busy here. So I will do this now. I will go to configure this page, and then the reset configuration button at the bottom to reset my page to the original configuration. And I will do the same for the track order, which is just here. And close the menu. You can find the same just above the picture as well, so these two icons here. Yeah, this is what I wanted to show on this page. Are there any questions on this demo? Anything unclear? Want to see more? Yeah.
In the PowerPoint, you can select columns from down middle marks in red.
At the top, the marks in red, yes. In the chromosome, the question was about the these marks here in the chromosome picture. These are alternative sequences. There are two different types. Some are red, some are green. The green ones are the patches. There's one on the very right-hand side which is green. That's a patch, so that's an update to the genome assembly, which will be incorporated in the next version. And the red ones are also alternative sequences, but they are haplotypes, so they are real variations in that region, which will not be incorporated.
In variation because they are not SNPs, right?
They're not SNPs, no, they are larger regions in the assembly, which are different. Yeah. And you can actually compare those to a reference sequence, to the reference sequence, and I think exercise 2 is doing that. Yeah. More questions? Okay, if there are no more questions, then we can have a coffee break now. Do we need to go elsewhere for a coffee? Or?
So can you
El café vamos a tomar a un profesor de acá, al lado nuevo afuera, que no me sigue. Cuando vamos se llama los llaves, así que sentado en la cantina.
Lo estoy buscando. No sé qué es.
Capaz de
Ahí,
sacar.
está ahí.
Y, pero
No,
no,
todo eso, mira.
y no lo van a poder prender entonces.
Acá.
Sí.
¿Se me corre muerto, no?
No, no sé, preguntale algo para que corto la
Mucho frío hacia acá.
No, no, totalmente.